gene expression and cancer. The word cancer was first used by Hippocrates 2,400 years ago. He saw a similarity between the swollen veins radiating from a breast tumour and the legs of a crab. Cancer is a group of diseases caused by a damage to the genes that regulate mitosis and the cell cycle. This leads to unrestrained growth of cells. As a consequence, a group of abnormal cells, called a tumour, develops and constantly expands in size. Cancer is a common and destructive disease. That being said, cancer is to some extent avoidable and, if diagnosed early enough, successfully treatable. Types of tumour. Not all tumours are cancerous. Those that are cancerous are called malignant, while those that are non-cancerous are called benign. Characteristics of a benign tumour include: it can grow very large, it can grow slowly, the cell nucleus has a relatively normal appearance, cells are often well differentiated and specialised, cells produce adhesion molecules that make them stick together and so they remain within the tissue. They form primary tumours. Tumours are surrounded by a capsule of dense tissue and so remain in a compact structure. They're much less likely to be life-threatening, tend to have localised effects, and can usually be removed by surgery. In contrast, malignant tumours can also grow to a large size, but grow rapidly. The cell nucleus is often larger and appears darker due to the abundance of DNA. Cells can become undifferentiated or unspecialised, and they do not produce adhesion molecules, so they can spread or metastasise, forming secondary tumours. They're not surrounded by a capsule, and so can grow finger-like projections into the surrounding tissue, making them very difficult to remove. They are more life-threatening because the abnormal tumour tissue replaces normal tissue, and they often have whole-body effects such as weight loss and fatigue. Removal usually involves radiotherapy and/or chemotherapy as well as surgery, and they more frequently recur after treatment. The development of secondary tumours from a primary tumour occurs in the body, and this is called metastasis. Cancer and the genetic control of cell division. DNA analysis of tumours has shown that, in general, cancer cells are derived from a single mutant cell. The initial mutation causes uncontrolled mitosis in the cell, and later a further mutation in one of the descendant cells leads to other changes that cause subsequent cells to be different from the normal in their growth and appearance. The two main types of genes that play a role in cancer are tumour suppressor genes and oncogenes. Most oncogenes are mutations of proto-oncogenes. Proto-oncogenes stimulate a cell to divide when growth factors attach to a protein receptor on its cell surface membrane. This activates genes that cause DNA to replicate and the cell to divide. If a proto-oncogene mutates into an oncogene, it can become permanently activated because the receptor protein on the cell surface membrane can become permanently activated, so cell division is switched on, even in the absence of growth factors, and the oncogene may code for a growth factor itself, and that production in excessive amounts stimulates excessive cell division. Both of these result in cells that divide too rapidly and out of control, and a tumour or cancer develops. A few cancers are caused by inherited mutations of proto-oncogenes that cause the oncogene to be activated, but most cancer-causing mutations involving oncogenes are acquired, not inherited. Tumour suppressor genes. These slow down cell division and repair mistakes in DNA. They tell cells when to die, called apoptosis. They therefore have the opposite role from proto-oncogenes. As its name suggests, tumour suppressor genes maintain normal growth rate and cell division, and so prevent formation of tumours. If a tumour suppressor gene becomes mutated and inactivated, it will stop inhibiting cell division and cells grow out of control. The mutated cells that are formed are usually structurally and functionally different from normal cells. While most of these cells die, those that survive can make clones of themselves and form tumours. There are a number of forms of tumour suppressor genes, including TP53, BRCA1 and BRCA2. Some cancers are caused by inherited mutations of tumour suppressor genes, but most are acquired. For example, more than half of human cancers display abnormalities in TP53. Acquired mutations of the TP53 gene occur in many cancers, including lung and breast cancer. The p53 protein is involved in the process of apoptosis, which is programmed cell death. This process is activated when a cell is unable to repair its DNA. If the gene for p53 is not functioning correctly, cells with damaged DNA continue to divide, leading to cancer. An important difference between oncogenes and tumour suppressor genes is that while oncogenes cause cancer as a result of activation of proto-oncogenes, tumour suppressor genes cause cancer because they are inactivated.